I'd like to introduce the next speaker. Professor Zheng is here. He goes by Jim. I'm really thrilled that he was able to make some time. He's professor of bioinformatics in our School of Biomedical Informatics, the McWilliams School of Biomedical Informatics, first school in the U.S. that's dedicated to bioinformatics. And the point where we're at really is I wanted to make sure that we had to talk about the benefits. What are the benefits for AI in healthcare? His work and his background really lends to a great topic to think about how AI is going to be used to think about drug discovery in the future. Thank you.
Thank you very much for having me. So I'm going to talk a little bit more technical, but I will try to be as general as possible. So in my school, we have over 50 faculties. Many of our faculties have this cross-disciplinary background where, one side is more technical, like computer science or statistics or mathematics. And the other side is the like medicine or biology. So many of our faculties have this kind of dual background. That's the same for me as well. So I have training PhD. My PhD is in molecular biology and biochemistry, and then my master's degree in computer science. And then I work in the industry for many, many years, develop algorithms and software analyzing biological data, and then come back to academia and work with many people developing new methodologies. For the biomedical data analysis, as well as building computing infrastructure to support, the biomedical research. So, as many of you probably aware of, for the current AI revolution, right? One important aspect is the computing infrastructure. Many of you probably heard about, they're going to build a big data center here or there, or, there are also controversy about whether they drain all the electric power or they will impact the local economy, all those kind of things. But at the core of the current AI revolution is really the computing. So, on the biomedical side, we try to incorporate all these new advancements in data science and AI to help with the biomedical data analysis and understand the disease mechanisms and the things like that. So as you may know now, the biomedicine really advanced very fast. One aspect of that is, from all these presentations, you can see that, investigation now is at a multi-scale. We have molecular level, we are looking at the small molecules as drug to treat the disease. We also have investigations at the cell, subcellular level or protein level to understand the disease mechanism. And we also have like imaging, to look at the brain image to understand the stroke and other disease. And also, like at the individual level, we have electronic health records to understand, the patients when they visit the office, what are their symptoms, signs, and the diagnosis, and then, to use that information for, clinical decision support. And we also at the population level, we look at the entire population, looking at, you can see that from Framingham study on, the cholesterol and how that might impact the mortality. So all these different level of studies in the old days, it's more like an individual. People study one level by one group of people, study another level by another group of people. But with the electronic health records that is available to many people, and with all these data sharing policy from NIH, now we have access to all kinds of data. For example, one study we have information for individual patients from the EHR data. But we also collaborate with the clinicians to collect the blood sample, to do the sequencing, to study their, blood cell, like immune cells and, proteomics and all those kind of things. And then we try to integrate all of them to predict the treatment outcome. So So that's why, nowadays, we generate all these data as fast as we can, and then we try to integrate them. But the significant challenge is really, how can we do that? Do we have enough computing power to do that? Because all these data from different, measurement, they different in all different ways. There could be batch effect, there could be, the confounding factors, how to eliminate all those challenges and make sense out of it. So that's where we now try to leverage data science and artificial intelligence to analyze all these data to really gather the most out of it to help with the clinical decision support, with the disease mechanism understanding, and all those kind of things. We started with these on cancer, but now we really generalize that to many, many different diseases, like autoimmune disease, the brain disease, as well as things like a stroke. So we actually realized the importance of this computing infrastructure, and we started to build what we call the modern computing infrastructure to support data science and artificial intelligence. And two examples here. One is this bioinformatics and high performance computing service center that I started in 2015. And the other is data science and informatics core for cancer research we established in 2017, thanks to the separate funding. So we build all these computing infrastructure and then try to really advance the biomedical research. So for those who are not really kind of the computing savvy, or it may be too technical, here, why do I have this? Okay, all right. Anyway, so basically, for the modern computing, right, we have two types of microprocessors. One is called the CPU, which is a central processing unit. This is a very versatile general purpose processor that is in all the computers you're using on your phones and on your desktop and things like that. And they handle very complex logics. It's flexible, but the challenge is that they work on the problems step by step. Okay. So on the other hand, there is this what's called a GPU graphic processing unit. So I have to say things from my memory now. But this GPU is not that new. In the old days, people developed that, but the capacity is more limited. Now it's very powerful. So this graphic processing unit, the one feature is that they have many, many microprocessors that they can process all the data in parallel. So instead of you have a sort of a pipeline to process data, you can take large amount of data, put on these graphic processing units, and then they have many, many different microcores to process the data. So by doing that, they have become very, very efficient, and they can process a lot of data at the same time. And also, there's another important aspect of that is that these kind of microprocessors, they are very good at massive matrix operation. And so, mathematically, a matrix is really a way to capture and represent the complex data. And but it's very difficult to process the huge amount of matrix that is what at the core of the current AI revolution, actually. So by using the graphic processing unit, now we can really process massive amount of data through the matrix operation, and that can be used for deep neural network and transformer models with billions and trillions of parameters. That's what all these chat GPT and other AI tools that based on. And also like image recognition, the chatbot, and the recommendation system, all these advanced AI systems really depending on these GPU microprocessors. And so the latest NVIDIA Rubin GPU, which is coming out this year, they offer roughly 50 petaflop of dense FP4 compute. So that's kind of very technical, but basically they are very, very fast. Okay, so one of these microprocessor really is as powerful, even more powerful than the huge the computers that in the old days like IBM has, that occupies the whole room. So these technologies getting faster and faster. For those who buy the NVIDIA stocks, they make a lot of money. I think many people knows that. So, realizing the importance of the computing infrastructure, here's what we built. We, in the past several years, I spent about $2 million from, grant funding, different resources. We built these very comprehensive computing infrastructure that has a large memory server with 6 terabytes of memory and several of these advanced GPU servers that we can train the large language model, and we can run these kind of ChatGPT like models and to analyze biomedical data. So with this kind of robust computing infrastructure, then we have been using it for many, many different biomedical research projects. One example is we try to use this GPU microprocessor to accelerate the clinical data analysis for the prediction. So basically, we try to collect the clinical information, the EHR data, from a database that has information for over 70 million patients. And then we sort of identify the patients with the specific traits. In this example, it's like the liver cancer risk in the non-alcoholic fatty liver disease patients. And so we identified over 100,000 of these patients. And then we try to build an AI model to predict what's the risk of a patient that has this liver cancer in the future. So what we developed is called this dynamic disease risk prediction. So it's nice to have some music. Yeah. So basically, the idea is that we have the electronic health record for these patients, and then we develop this model, and then we can make a prediction, say, well, how big is the risk of these patients for, say, stroke? And then the patient might have a new visit, and then the new information is added to the electronic record. The key here is that how can we incorporate this new information to update the model and make the prediction based on the past information as well as the newly added information. So this is called a joint modeling for dynamic prediction. And so one challenge of this particular prediction is that because you have a lot of patient information, and also you have the dynamic information, you keep adding new information. So making the prediction would be really, really challenging, and it's very computationally demanding. So what we did is that we try to leverage this a massive processing power of the graphic processing unit, and then to accelerate this prediction. So in the end, our methods can predict, make the predictions using these massive amount of data, over 100,000 patients, to train the model and to make the prediction, where the current other models, they cannot handle this massive amount of data. So this is as an example to show you that how we can use the GPU to accelerate, the biomedical prediction. So the other project I'm working on is NIH Brain Initiative. So the idea is try to use all these new technology to understand the human brain so that we can better treat the disease like stroke. And so for this NIH Brain Initiative, what we started is to sample the brain cells from different part of the brain and try to understand how many different kinds of brain cells in a human brain. And also we also investigate the mouse and other non-human primate brain as well as animal model to develop the treatment strategy and other things. So for this project, we basically focused on the human and the mouse brain. And the Brain Initiative project sampled over 4 million brain cells from the mouse brain and over 3 million cells from the human brain. And then they use a technology called single-cell sequencing. The idea is to use this technology to understand for each individual cell what kind of genes are expressed in these different cells. And then based on their gene expression signature, then we cluster all these cells into different groups. So for example, you have maybe 100 cells, they have the gene expression, similar gene expression. They have gene A is all expressed, the gene B not expressed, and all this kind. Based on this pattern, we cluster all these cells together, and to our surprise, we have identified over 5,300 different cell clusters, where in the old days, neurologists, based on the morphology, based on the physiology of the neurons, they only assume that, there could be about, a few hundred different type of brain cells. So that is a big surprise. Now, the challenge becomes, what are these 5,300 different brain cell types? And that's where we start to use the AI models to solve this problem. So the idea is that we take an AI model like a chat GPT-like model, and then we want to use this kind of model to help us to understand that, if this cell type express this group of genes, what this group of genes is doing and how they help us to define what this particular brain cell type is. And so we started with these signature gene sets for each brain cell types. And then we ask these models and give us functional description and also, some predefined nomenclature to describe their function using what's people call ontology. And so this kind of approach basically leverage the enormous power of these modern AI models. I believe many of you probably use the chat GPT, right? It's really amazing. You ask a lot of questions, they give you answers, they can do a lot of things we cannot do before. So what we try to do is try to leverage its power so that we can better understand the brain cell function. So obviously, when we use these kind of models, by the way, this is actually generated by ChatGPT, and you probably can see the pattern, right? But so there are two. I'm asking, well, if I use this kind of model, what's the limitation? So ChatGPT actually giving me some honest answer on this. So there are two limitations on using these kind of models. One is that these models, they are trained on all the data we collected from internet, from books, from everything, and they are not specifically trained like a biologist, right? So you can ask them a lot of questions, but when you want to expect them to give you answers like a PhD level biologist, they cannot do that because they are not trained like that. It's just like, if you really want a neurologist to give you some right answers, they have to go through the particular training programs to be able to do that. So that's the first challenge. The second challenge is the hallucination. That is, as we all know, when you ask ChatGPT, a lot of times they could hallucinate. They give you answers that may not be correct. So we need to also be careful about that, especially when we use that for scientific application. When we try to make decisions, we want to make decisions based on the made-up information by these kind of models to treat patients. So To do that, basically, we developed this AI agent-based system to analyze these brain cell marker sets and cell types by overcoming those two limitations. So the first limitation, for the first limitation, what we did is that we fine-tune this model. So, like I mentioned, for the ChatGPT, they are trained on the general text. But what we did is we collect a set of biological data that is well analyzed and understood by, biologists. And then we took a general AI model, and then we teach them about the biology using this data. For example, there may be a gene set on, related to stroke, and then we give them this group of genes and ask them to generate to describe what this group gene is related to, and it'll give an answer. And then we compare that to the ground truth. Say, oh, this group of genes supposed to be related to stroke, and then we calculate the difference, and then we then through these what's called a back propagation, we modify the parameter of these models so that next time they will give a better answer. So by doing that, it's sort of like we now push this general AI model through a graduate program for biology so that they understand better about the biology. So the second thing we did about the hallucination is that we require the model only synthesize information using the provided literature. So after our initial fine-tuning, and we use these AI models to generate the description of these cell types and the marker genes, and then we use that generated information to identify the scientific literatures related to what they initially think the biological function of this group of genes and cell types. But then we ask the model to refine the final description of the functions of these genes and the cells by only using information from this literature. So by doing that, what the model generated is all supported by the scientific research that's published. So that way, the information, the final information generated is not hallucinated, it's not made up information. Rather, they are linked and supported by the published scientific literature. So by doing that, we basically annotated over 20,000 brain cell marker genes and over 5,300 brain cell types for mouse. And also we did that for over 3,000 brain cell types for human. So in the end, we have this resource that we put on the internet, and everybody can look up, if they're interested in particular gene or particular cell type or particular disease, they can search and they can identify what type of cells might be related, to the things you are interested in. And if you take a picture of the barcode, and you can, go to our website, and you can sort of find out. Besides these, we also worked on the the other aspect of biology. For example, alternative splicing. Human genome has over 25,000 genes, but we all know we are pretty sophisticated, right? How 25,000 genes can help us really dealing with all come up with these sophisticated beings. And one way is that through a mechanism called alternative splicing. And that is, you have one gene, but our cells have a way to make a different copy of these genes through this what's called alternative splicing. So they have a different version of these genes, and each version may have a different function. So that's why, even though we only have 25,000 genes, but overall we have much more different version of proteins that can be encoded by these 25,000 genes. So we use an AI model called AlphaFold to predict the protein structure, and then we compare the alternative spliced form of this protein to the original form of this protein that generated by the gene, and see how the structure, these different version of these genes can is impacted by the protein structure, and then we connect that to the disease and understand how things work. So now with all these AI tools, we certainly can advance more on the AI-assisted drug development and screening. So as I mentioned earlier, one of the AI breakthrough is to predict the protein structure from the primary sequence. As many of us know, that the function of a gene is really executed through the protein, right? So understand the structure of the protein is a very important step for us to understand the disease mechanism as well as for the drug development. So basically, AI tools like AlphaFold with the powerful GPUs we have can take primary sequence and make predictions of the protein structure. We specifically use that to predict the protein structure for antibodies, which, as you may know, a lot of these, nowadays therapeutics is based on antibodies. So when we make these kind of predictions, we certainly want to know, how the quality of the prediction, whether they're reliable, and whether we can use it really for the downstream research and use different metrics like IPTM and the PAE. And we did an in-depth analysis of using these AI models to predict the protein structure, and also evaluate how reliable they are and whether they have the hallucination and how to solve the problem. And one example is this protein called parvalbumin, which is a biomarker for the stroke rehabilitation. And for this protein, it's important to develop a reliable antibody to see, where they are, to detect, how they change during the stroke recovery. And so we use this AI model, develop, evaluated antibodies against parvalbumin, and try to identify which part of the parvalbumin protein this antibody binds. And we identify like 2 different epitopes, and we did, the mutation, virtual screening, the mutations, to confirm this prediction of different epitopes. And so that we can have good antibodies for the detection of this protein and for many, many different applications. So the other thing is also when we perform the therapeutic development, we want to understand how the small molecule as well as the large proteins bind to its target. And this is through an approach called the molecular dynamic simulation. And we also use that for the virtual screening. So basically, the idea is that we have a digital version of these proteins and the small molecules that bind to this protein as a potential target. And then we try to use some kind of energy form to see how these small molecules bind to these large proteins, and what are the residues that might be important for this binding. And through these kind of molecular dynamic simulation, we can have a really good understanding of these proteins and its potential therapeutics at the molecular level. And this is also enabled by the GPUs I talked earlier. In the old days, when you try to run this kind of simulation for a very short period, it takes very, very long, take days or months. But with the powerful GPU, we can run very, very long period of the simulation very fast. So that really enabled us to have a comprehensive evaluation of these small and large molecules that target the proteins that's of interest for, say, stroke treatment. And, here are just some, energy landscapes through, these molecular dynamic simulation, which allow us to understand how, the protein interact with the antibodies target too. So the other thing is for the small molecule, we also leverage these powerful GPU servers to, do the virtual screening to identify the potential small molecule that can target the particular target. So the example I use here is a small protein from the bacteria that, we use the this virtual screening, use the drug library from the drug bank, and we analyze the target, try to identify where are the potential binding pockets on this protein. And then we, after identify those pockets, and then we use the small molecule library that has millions of different small molecules. And then we try to, match them between the targets, the pockets we identified, and each of these small molecules. Again, the powerful computers allow us to do this very fast. In the end, we identified 2 out of the top 20, so we after our screening, we pick the top 25 candidates, and then our collaborators in the lab, they evaluate whether the candidates we provided really bind to the target. And the 2 out of the 25 top candidates indeed bind to the target. So which really give us, a high confidence that now we can run this virtual screening very efficiently to identify the potential targets for the drug development. And here's just like a movie for the target candidates we identified, shows how it binds to the target protein and, how dynamically this protein sort of change over time. And again, this, is really because of the powerful GPU servers we have. And in the end, I think, I showed you some examples how, the robust computing and the modern data science and AI technology can really advance, the biomedical research and the drug development. And hopefully we can sort of collaborate to work in the area of the stroke treatment and prevention. With all that, I will just stop here.
Thank you, Dr. Zheng. So we'll hold the questions so we can just have one more speaker.